Hey guys, it's Mr. Thomas, and I wanted to go through with you a little bit on chapter 6, which is all about cell communication. Now that you've had a background in a little bit of cell biology, learning what a cell is, many of the main parts and functions of those parts inside of a cell, now it's time to, and you've also covered transporting items through the cell. Now it's time to learn a little bit about how cells communicate and how they send information or chemical signals to other cells. So we'll start here with communication between cells. This is very important to help cells maintain something called homeostasis. Now, one thing about really all of the chapters, but also this chapter that is very important, is to find some way to study and learn the vocabulary. There's a lot of vocabulary words, especially in this chapter, that deals with cells and cell communication. So find some way that helps you understand, learn the vocabulary. I would suggest writing down the vocabulary. I know a lot of students use the Quizlet app. I would suggest making your own flashcards. There's something about writing these vocab words down that really helps you remember those. So write the terms down on your own. Maybe make a vocab journal or at least flashcards to help you memorize these terms. So first of all, that homeostasis, I think we've seen before, homeostasis is that equilibrium or balance that is very important for all living things. In humans as well, we have to have a balance internally. And so in order to maintain that type of balance and equilibrium, cells have to be able to communicate with each other. And so we'll talk about that kind of throughout the chapter. Now, to give you an example here of cells, this is, well, first of all, maintaining homeostasis in multicellular organisms, but also in single or unicellular organisms such as bacteria. Communication is also very important there. And they give you an example of something called a biofilm. That's just where bacteria kind of organize and group themselves together, almost like in a thin layer or sheet. And it requires a lot of coordination and a lot of communication for bacteria to kind of organize in that type of fashion. Now, the other thing is we'll talk about this as well. Organisms, even of different species, use chemical signals to communicate with each other. And you don't always think about things like bacteria and seaweed having some type of communication, but there's actually evidence that shows that bacteria and a particular type of green seaweed called Enteromorpha actually can communicate through various types of molecules, and they actually live together in a symbiotic type relationship. We're going to start out talking about cell signaling, where cells are communicating with one another, like inside of a multicellular organism. And there's four main processes that your book talks about. They're listed there for you. And we'll kind of start with these: signal transmission, reception, signal transduction, and then finally a response. So let's look at these as we go through. If you're following along in the textbook, I'm on page 132 in chapter 6. Signal transmission is what it sounds like. You have to get some type of signal, and usually this is some type of chemical, some type of molecule type signal that is produced or synthesized through a cell, and then it has to be transmitted to some type of nearby cell. A lot of times when I see this type of information, I think about how your human body, how our human bodies transmit hormones. Hormones are chemical signals that are produced by a type of gland cell. They get released, and then they have a target cell, usually close by. If it's not close by, then it has to be transported. And again, for the human body, those signals must be transported through the blood. You also have to create that signal using some type of chemical. Usually, you have to be able to receive the signal. That's reception. And so you have to have some kind of receptor. Usually you find a receptor somewhere either on the cell membrane, or sometimes there's an internal receptor. That receptor is usually what's called a glycoprotein. This is just a type of molecule which is mostly protein, but it does have a carbohydrate attached to it. And so the important thing about these receptors is that they have the ability to bind specifically with whatever signaling molecule you're talking about. And so it's like a lock and key type scenario where that receptor has to bind specifically to the molecule, to the signal molecule, otherwise there's no reaction. Then the last two steps there, signal transduction and then response. Signal transduction means that that signal, that chemical signal is converted to some type of cellular information, right? So the cell converts that signal that we just sent, it gets binds to a receptor, then that cell has to transduce or convert really that chemical signal into some type of signal that the cell can respond to. And so we'll see some examples of this. There's one called a G protein complex, and I'll show you kind of some examples of that as we go through the chapter. But it just involves converting the chemical signal into a signal the cell can use. And then you have some kind of response. That's the last step. That converted signal will have some kind of cellular response. You know, a lot of times it is, they list here in the notes here that a lot of times it can open and close a plasma membrane, or open and close a protein channel along that plasma membrane. It's like open and closing a door. It could cause the cell to respond in like a growth type fashion, such as growth hormone can cause cells to grow faster, or can increase the cell size. So there's lots of different ways. It could cause mitosis to increase or decrease or whatever. Section 2 is going to briefly talk about examples of how cells do communicate. Now, we've already seen how cells can communicate through cell junctions, and there's a note here, you can go back and look at chapter 5, how cells have various cell junctions, gap junctions especially, that you find in cardiac muscle cells that can transmit signals, electrical signals, very, very quickly through through cells that are very close together. They can also communicate through electrical signals. And in the diagram here, the picture, this is, if you're following along in the book, this is on page 134. You've got a few diagrams here. This shows two nerve cells, two neurons, and their signaling molecules are shown here. These are basically neurotransmitters. So you have one neuron here. Here's the cell body. These are dendrites coming in. Here's the axon coming out. And then they're releasing neurotransmitters into some type of junction here. And they are picked up by the next neighboring or the target neuron in this case. They have receptors on the ends of the dendrites. They pick up the signal, and then they would respond. So electrical signals are very important. You may have a signal communication cell to cell that's just based on direct contact. The cells actually have to make contact to each other. And so you have kind of a example here in the picture here, where you've got one cell here that has a particular chemical that's already bound to this cell's receptor, the little blue area here, that is a receptor here, and it has some type of chemical signal bound to it. And in this case, in this example, the cell would come in close proximity with another local cell and actually transmit its chemical signal directly to that neighboring cell. The third example here is really a hormone type of example. In other words, you've got a cell here. This would be like an endocrine cell, a gland cell producing some type of chemical secretion that would be deposited and would diffuse into a blood vessel. I know it's kind of strange looking, but this is supposed to be a blood vessel. And so you have chemicals going into the bloodstream where they diffuse, and they find a target cell somewhere else that they have a specific receptor for. So that's kind of how they transmit. Hormones are transmitted through the bloodstream to the target receptor. This slide is just kind of giving you some examples of some of these chemicals that are produced. You don't necessarily need to know all the different examples here, but some cells do produce local, what they call local regulators. These are just chemicals that don't travel very far. They help, they activate nearby cells. These are some examples: growth factors, histamine. If you have any kind of allergies, especially in the springtime, you may have to take an antihistamine. Histamines are produced and as a chemical that kind of start up the inflammatory response. And so a lot of people have problems with those. Nitric oxide, that affects your, a lot of times it will affect your blood vessel dilation and things. And then something called a prostaglandin. These are similar to hormones. They're a special type of hormone that can have the effect of, again, changing your dilation of your muscles, or relaxation of your muscles, including inside of your blood vessels, which causes your blood vessels to dilate. Now, section 3, we'll talk a little bit about reception. We got the cells. The cells are producing some type of chemical signal, and so now we got to get that chemical signal has been, released, and it travels maybe, or it may be a local chemical signal. Now it has to bind to some type of receptor, and so remember, this has to be a specific type of receptor, has to bind to a specific molecule. Here's a good vocabulary word to know. It's called a ligand. It's a little strange of a word, but a ligand is essentially some type of chemical that will bind to a receptor and cause some type of response. Here's a couple other vocabulary words: hydrophilic and hydrophobic. Hydrophilic type of chemicals or molecules. These are what are, kind of break down the word. This means water loving. What this means is that these molecules bind to their receptor on the surface or on the membrane of the target cell. That's because these are water soluble. That means they don't go through the cell membrane, right? So the cell membrane, if you remember from the earlier chapter, the cell membrane is mostly lipid. It's a lipid bilayer. So a water soluble molecule is not going to penetrate or diffuse through that lipid bilayer very easily. Some water does, but not very much. And so if the hormone or if the chemical signal is hydrophilic, then it needs to have a receptor on the surface of the cell. A hydrophobic molecule is essentially a lipid-based molecule, which means it can diffuse through a lipid membrane, which means its receptor can actually be inside of the cell or intracellular. And this slide here just kind of gives you some examples of some ligands and receptors. It also mentions, again, that only the signaling molecule that actually fits the shape of the receptor can trigger response. And again, that goes back to the specificity. They have to match. If a chemical signal would happen to, come across a receptor that it didn't match, then there wouldn't be any kind of response. And so you've got different cell types making different types of receptors, many different types of receptors with different, what are called active sites on that receptor would be there. These are this slide lists a few examples, like rhodopsin. You may have seen that before. That is a pigment that's located in the eyes, in the nerve cells in your eyes, that helps to pick up and help your eyes detect like light and dark. Phytochromes, these are chemical signals that are found mainly in plants, and so are cryptochromes as well. Now, there is something called upregulation and downregulation. This helps to, as it sounds like, kind of increase or decrease the sensitivity of the reaction. And so the process of upregulation is going to give you an amplified type of a signaling. Even though you're making the same amount of hormone, you basically make more receptors. And so having more receptors, you generally have an abundance or a higher amount of hormone than you would actually be able to use. So if you have more receptors, then that allows you to amplify the signal. without having to make more hormones or more chemicals. Downregulation is kind of the same thing. If you need to decrease the reaction or the cell's response, one way to do it is to decrease the amount of chemical signal, but you could also decrease your cells, also decrease the amount of receptors. And if you drop the number of receptors, then you're going to downsize the response. And so your lysosomes that we've talked about before that help, they contain the digestive enzymes inside of there. They can actually digest some of those receptors so that you have a lower response. Now, there are different types of receptors that you'll find. And we'll kind of look at 3 examples. A lot of these are what are called ion channel linked receptors. In other words, they're ligand gated. There's that word ligand again. In other words, these are receptors that they have a gate across, and they only open their gate if the ligand is present, if that chemical molecule is present. And so here's a few things about these. These are found in the plasma membrane. They're a receptor that's bound, kind of internally into that membrane, but they have a portion that is kind of sticks outside the cell. They have the ability to convert chemical signals into electrical signals, and the ion channel will either open and close, depending on whether or not that ligand or that chemical molecule is actually attached to it. And a good example of this is something called acetylcholine. This is a type of neurotransmitter. You find this a lot of times in muscle cells or muscle physiology. And so whenever that acetylcholine is present, it will bind to a ligand-gated sodium channel. And so if the acetylcholine is present, it'll bind to that channel, and that causes the ion channel to open. It's basically opening up a pathway or gateway that allows sodium then to go into the next cell. And so your textbook has a few examples here of these. There's a nice set of figures, figure 6-5 on page 136, that shows you some examples. And here you see an example of an ion channel linked receptor. In the picture here, I can see my lipid bilayer membrane here. I can see I've got this receptor that's bound, internally here. Its active site is on the outside of the cell. Here I've got some kind of signaling molecule. This may be like in the last example, the acetylcholine. I've got some sodium ions that are kind of stuck outside, and the reason is because it's basically this ion channel here is closed, it's gated. And so in the next slide, you see where the acetylcholine or the chemical binds to the receptor, opens it up, and that allows the channel to open. Now my sodium can diffuse across that, goes through facilitated diffusion and can go from the outside of the cell internally. Now, I mentioned this earlier. This is what's called a G protein, the second example of a receptor you'll find, G protein linked receptor. We'll look at the picture of that as well. So let me kind of read over a little bit of this, and then I'll kind of explain it, showing you the picture a little bit better. You have, again, some protein receptors, transmembrane proteins. They have a binding site for a signaling molecule on the outside. That's just your general type of receptor, just like we said before. But in this case, it also has a binding site on the inside of the cell, and it binds to something called a G protein that goes inside of the cell into the cytosol. And there's other signaling molecules that will create a pathway, a biochemical type reaction, almost like a domino effect inside of the cell that is linked to this reaction. So here we have our same membrane set up. In this case, you've got a little bit more complicated protein receptor here, but it's basically a receptor that will bind to a signaling molecule. The molecule comes in, it binds to the receptor. In this case, instead of opening a channel, what it does, there's what's called a G protein that's embedded inside on the inner portion of that membrane. Inside, this would be inside the cell where the cytosol is. And so this is the G protein is bound here. This creates a reaction. Once this receptor once the signaling molecule binds to the receptor, it creates a reaction with this G protein. And now it kind of links up with that receptor. and that basically turns on a neighboring enzyme. So here you have a protein enzyme that's embedded in the cell membrane as well. And so all of this basically activates this enzyme. Now, enzymes, as we've seen before, they're biological catalysts, and they help to kind of speed up some type of reaction. This would speed up some kind of reaction to make the cell have some kind of response. Then the enzyme-linked receptor, this is similar to a G protein type format. You still have a transmembrane protein, just a protein receptor that goes through the membrane, is what that means, has a binding site for a signaling molecule outside the cell, just like before, but this is linked to an enzyme directly inside of the cell. And a good example of this is how insulin works and some other growth factors work. And so let's look at this this example here. On the left here, you see, again, you've got your protein-based receptor. It's got an active site outside. It's transmembrane. In other words, it goes all the way through the membrane. You've got the signaling chemical molecule here. And then linked to this receptor, you have a series of enzymes on the inside of the cell and the cytosol. And so these basically will, again, activate once the signaling molecule attaches, binds to the receptor. Now on the right, you see that these enzymes here become activated. That's the big thing here. They become activated. Now, one thing to note here is this does require energy to work. This ATP here is the energy unit here that's required to get all of this activated to work. And once this activates, then the cell could have some type of response. And again, the example that they listed on the last slide was how insulin binds to a receptor and will eventually cause some type of protein channel to open that allows glucose to leave the bloodstream and go into a cell such as a muscle cell or the liver cell to try to get that glucose into a cell where it can be used to create energy. Now, so far we've been talking about receptors that are located in the membrane. So we're talking about receptors that are essentially extracellular receptors. That's because they need they have hydrophilic type molecules or chemicals. But remember, you could have a hydrophobic type molecule that could actually go inside of the cell. It could diffuse through the lipid bilayer. And so sometimes you have intracellular receptors that are found inside of the cell, either kind of free floating in the cytosol or the liquid portion inside, or even in the nucleus. And essentially they work as what's called a transcription factor. And we'll get to those a little later, but these are essentially proteins that help to turn on a gene. They actually turn on a genetic sequence. And so again, these molecules diffuse across the membrane, and they'll find their receptor either in the cytosol or maybe even inside the nucleus. They form a transcription factor. They bind to the DNA and essentially turn on that particular gene sequence. Section 4 is going to look at a little bit about the next step in the process. So far, we've got the cells that have secreted the chemicals. We've transmitted that signal. And now this is the signal transduction. This is getting that signal converted to something that the cell can actually use. So for signal transduction to take place, you have to have those regulatory molecules, some type of chemical signal to bind to the receptor that activates those membrane proteins, and then that will lead to a series of chemical reactions that will transduce or convert that signal into something that the cell can respond to. And so we'll kind of go through a few steps. That signaling molecule, that chemical, will bind to some kind of cell receptor. Again, it generally involves some type of changing the shape of the receptor tail. That's the part that goes down into the cytosol or the cytoplasm. Remember, we're dealing with protein receptors, and so generally protein receptors a lot of times can change shapes. They change the conformational shape of the protein whenever some kind of molecule or ligand binds to it. And then that signal then gets relayed through a set of chain reactions, essentially. These are called protein kinases. There's a figure 6.6 that's in your textbook. I don't have a copy of that one on the slideshow here, but I do want to show you something similar to that. It essentially creates a signaling pathway that helps to increase or amplify that signal inside of the cell. And so let me show you a little bit about this. What we're dealing with here, first of all, is that type of enzyme-linked receptor. This is where it all begins, right? You've got this signaling molecule, your ligand, it binds to the receptor. It does require energy to complete this, like we said before, and then it will start activating what's called a protein kinase area here and start to change kind of the shape of this protein receptor on the inside of the cell. Now, this works kind of like an on and off type switch. Each part of the pathway, and I'll show you again, I've got a diagram figure that's similar to what's in your textbook on page 138. I'll show you that in just a second. But you basically have almost like a cascade, what they call a cascade effect, or it's almost like a domino set of chain reactions. Well, each of those reactions will either be an active molecule or an inactive molecule. The active one is considered to be on, and that's going to continue the amplification process. If it's off, then it doesn't continue amplifying the signal. And so basically they're referred to a lot of times as molecular switches. They have to either be turned on or turned off. And so what we're talking about here is the protein, what's called a protein kinase cascade. And those protein kinase molecules become activated whenever you add phosphorus to, or phosphate to, that group. And so that term phosphorylated or phosphorylation just means that they add, they use a enzyme called protein kinase to add the phosphate group to the group, to the molecule inside. And that uses this enzyme called protein kinase. Now, you have that would cause it to become activated. So remember, you have to deactivate or turn that switch off as well. So there's another enzyme called a phosphatase that actually removes the phosphate group. So if the phosphate group is attached, then it's activated and it's turned on. If you use phosphatase, that removes the phosphate group through a chemical reaction called hydrolysis, that removes the phosphate group, and that turns the pathway off. So these two enzymes are basically helping to control or regulate this whole pathway. All right, so here is the type of a flow chart. It's showing you the cascade reaction. This is just from a different textbook I found, but you have one similar to this on page 138. But we're not going to go through all of this in detail, but essentially you have a series of reactions. This would be the cascade. Notice that you only have one signaling molecule that is here. We'll go back to that in just a second, but here you have on the inside, this would be your protein receptor. On the inside here, you would have your inactivated protein kinase. Once this molecule here becomes activated, then it phosphorylates this molecule here, and now you have an active protein kinase, and that serves to phosphorylate the next one in the pathway. So now that this one is active, it can activate the inactive protein kinase number 2. And again, it does require energy to do this. And so now it phosphorylates this molecule, and now it becomes activated, and then that activates the next one in series. And so it just continues on. Eventually, you'll have some kind of cellular response. And again, it may be some type of growth of the cell. It could be that the cell opens a particular channel and allows glucose to come into the cell. This is basically how an insulin type hormone would work. But essentially, you are amplifying this signal. And again, we started out with one signaling molecule, but you have all of this pathway that essentially amplifies the signal until finally you have some type of response here at the end. And so that's one type of signal transduction reaction, is that protein kinase cascade. You also have a signal transduction using the G protein linked receptors that we looked at a little bit earlier. And this helps to regulate the transduction of in a lot of different types of cells. And so we kind of talked about this already. You have a G protein that's connected, it's linked to, on the inside of the cell membrane, and there is a molecule here of what's called GDP. This is like a low energy type molecule. This is similar to ADP. If you remember that, ADP is adenosine diphosphate. So this is guanosine diphosphate. This uses a different nucleotide base here. But this is GDP. This is like a low energy molecule. And then whenever you have the binding of the ligand or the signal molecule, then you actually have the GDP being replaced with the higher energy molecule, GTP. That extra phosphate group allows this to have higher energy. Then you have the GTP then uses its energy to carry on a reaction, and then that eventually deactivates that whole G protein. This part of this process, we didn't use this term earlier, but part of this process involves what's called a second messenger. That gets a little confusing until you realize that your first messenger really is just your signaling molecule. Whatever chemical binds to that protein receptor, that's really a first messenger. Your second messenger is going to be on the inside of the cell. And that's because, remember, that first messenger is most likely hydrophilic. The first messenger is water soluble, so it can't get through the inside of the cell membrane. And so you have to have some type of second messenger on the inside to relay that information. And so they help to amplify the signal inside of the cell, relaying that information to, inside the cell to some kind of protein or activate some type of enzyme inside of the cell to start some type of cellular response. A good example of a second messenger is something called cyclic AMP. And this slide here just kind of gives you a flow chart really of how this type of reaction would work. Here's your first messenger coming in, your signaling molecule. This is on the outside of the cell. It would bind to the G protein linked receptor. This is a protein receptor. It's transmembrane. It's going through the cell membrane. So your first messenger binds to it. That is linked to a G protein on the inside of the cell. It activates the G protein. That's going to activate an enzyme that helps to form that second messenger called cyclic AMP. And then once it's activated, it activates a protein kinase, just like we've seen before. This protein kinase, just like before, causes phosphate groups to be added. And so that phosphorylates other proteins, essentially activating other proteins in the cell, and that's what causes some kind of cellular response.